Hello, fellow scientists, and welcome to the start of our next review series. This particular review series is on your antibiotic resistance packet, and this video in particular will cover your antibiotic resistance lab and the lab summary. So it's going to be pages 2 through 12. So let's go ahead and get started. So the first thing that you'll want to do is pause and read this brief introduction, which talks about the idea of antibiotic resistant bacteria that they found in a cave. So once again, pause the video and take a minute to read that. So that brings us back to our actual lab. So it says simulating a bacterial infection investigation. And so what we're going to be doing in this lab is using a simulator in order to investigate how a population of bacteria evolves in response to antibiotics in some different scenarios. So the first thing that I want you to do is create a graph that represents sort of what do you think would happen, and this will be a line graph, to the number of bacteria if you were to take medicine and then stop taking it and then kind of start taking it again, right? And so for this line, you want to do something like how it would go down, right? As you take medicine, it decreases. And then you stop taking medicine, so maybe the bacteria increase. And then you can start taking it again until they all die. And that's just to show this idea that antibiotics is going to, hopefully, have a direct impact on the population of bacteria. And if you need to reread this is a slightly different scenario up here, but you can also do this one as well. But ultimately, you want to make a prediction about the interaction between antibiotics and bacteria and represent that with a line graph in some way. So then we have a hypothesis, right? So it says here, if you don't take your antibiotic prescription for the entire duration, so that's how you will start your hypothesis, then, well, make your prediction. What do you think will happen? So that brings us to our procedure. Now for this, if you're at home, you aren't really going to need to do the procedure. I'm going to have data for you, but I am going to demonstrate to you the tool that we used. So the tool that we had is right here represented on your screen. And what this does, this shows us a population of bacteria. So you can see we have some blue bacteria, which are the least resistant, fairly easy to kill with medicine. We have green bacteria, which are kind of normal bacteria, somewhat hard to kill, but not too bad. And then we have red bacteria. These are the resistant bacteria. These ones are quite hard to kill. And so what we did is we were basically looking at how many days does it take to kill bacteria over 3 different scenarios. In scenario 1, we take our antibiotic every day, which kills off some bacteria. They reproduce, and then we go to day 2 and we repeat that. We take some antibiotics, which kills off some bacteria, and we keep going until the bacteria disappear. In the second scenario, we don't take we don't take medicine in the beginning. So they grow and they grow and they grow. And then eventually we feel sick enough, we go to the hospital and we start taking the medicine. And then at that point, it's a lot like scenario 1, in that we're going to take the medicine until we kill off all the bacteria. And once again, I'll have data for this in just a minute. And then finally, the third scenario, and this is going to be the most important one for us. We take medicine until we feel better. So like the bacteria get down to maybe 5 or 10 bacteria, so we feel a lot better. And then we stop taking the medicine, which turns out is exactly what we don't want to do. And we'll look at why in just a minute. All right. So in your packet, you're going to have 3 data tables for those 3 scenarios. And you're going to have 3 graphs, 1, 2, and 3. Okay. So what we want to do is on Google Classroom, you'll find antibiotic resistance files. And then one of those is the simulator, and the other one is the Google Sheet. The Google Sheet has the 3 scenarios and the data that you need. So you can see here, scenario 1, here is the data. And this is essentially what your graph should look like, right? You can see the total population goes down over time. The easiest to kill blue bacteria die first, and then the green, and then the red. And so you can fill in the data table and then also create your graph. Now, your data table might only go up to 8. You can still graph the whole thing. Just fill in as much of the data as fits on your data table. Scenario 2, now this one you can see them getting very sick, right? So in the beginning, we get a rapid increase of bacteria. But then we start to take the medicine. Now, in this case, you might need to go to the hospital to get a stronger medicine. But it does work, and as long as you take all of it, it's going to kill off the blue, and then the green, and then the red. So same overall outcome. Now, scenario 3, this is the dangerous scenario. In this case, we take antibiotics until we feel better, and then we stop. Well, look at what happens here. In this case, what happens is, so we kill off the blue and the green. That's the least resistant and the normal resistant bacteria. And at that point, we feel pretty good. So we decide to stop taking the medicine. Well, and look at what happens. The bacteria that are left are the red resistant bacteria, and they are going to grow. Now, this is dangerous because now, if we try to start taking medicine again, it's not going to work very well because these are the resistant bacteria. They're hard to kill. So we've evolved from a population that was easy to kill, blue, to a population that is hard to kill, which is red. So we'll be coming back to this graph in particular for the questions. So once again, once you fill in your data table and graph, it says use the space below to describe the results of the graph and the experiment. Okay, so for that, you would talk about how scenarios 1 and 2 show that taking the medicine until the bacteria die is the most effective. Scenario 3 shows that if you don't take all of the medicine, you have a chance of evolving the population to become resistant. And once again, we see that in this picture right here, right? We see the red ones, they're the only ones left, and so they reproduce, and then that evolves the population to become more dangerous. So number 3 says if you stop taking antibiotics when only 3 red antibiotic resistant bacteria remained, what do you predict the bacteria population would look like in a few days? And is this good or bad? Well, that's exactly what we see in scenario 3, right? So if we only have red left, the red reproduce. This is very bad because these are antibiotic resistant, and therefore they're going to be harder to kill than the blue ones that we started with. So then this table is just summarizing each one altogether. So did it prevent antibiotic bacteria from growing? Scenario 1 was yes, we were able to kill them. 2 was yes, 3 was no. Right? Why? Well, because it says right here, antibiotic was taken every day, so that's why it worked. Here, once again, we although we didn't take it in the beginning, these scenarios almost become the reasons. Although we didn't take it in the beginning, we did take it until they all died at the end. This one, which was our no, did it prevent the growth? No, this one did not. And the reason why is that by stopping too soon, you're letting the resistant bacteria that remain grow, right? And so that's just another interpretation of those graphs. All right, so this question, it says, describe the limitations of this simulation to represent a bacterial infection. How could we improve it? Okay, the main limitation in our simulation was that even though we have different colors, they all are basically the same bacteria. We don't have any mathematics or any numbers or randomness within our simulation to really make the red ones harder to kill. We just killed them last in our simulation. So it simulated that they were harder to kill, but they weren't actually harder to kill. We could have made this more realistic and have numerically some mechanism for them to be harder to kill. So then it says, describe the limitations of the data collected and how it was analyzed. How could you improve the data collection? Okay, well, in this case, right, this wasn't data from like a real hospital. It was just a simulation. So this would be better if we used an actual data set from a health service. And we could actually look at, health data from a hospital that's dealing with these sorts of infections. All right, the last 2 pages are your rubrics. So these you can reflect on your own. The student rubric, this is your own personal score and your understanding of this topic. And then your partner rubric, that's for, I always say if you weren't in class for this, you can just go the last time that you were in class, reflect on how your table worked as a group and what were some strengths and weaknesses. With that said, we'll be continuing our conversation with antibiotic resistance in our next video, where we take a look at a claim of antibiotic use in agriculture.